Hey everybody, I'm Maya Cothran. I'm a senior at Howard University, and today I am going to be telling you guys about the investigation into the SPL7 orchestrated and microRNA regulated copper homeostasis and fertility in Brachypodium. So today I'm going to tell you all about the importance of copper homeostasis in plants, the model systems Arabidopsis and Brachypodium. Then I'm going to go into the role of microRNAs in the regulation system of copper homeostasis. And it's going to be twofold: developmental microRNAs and copper microRNAs. Then I'm going to go through my objectives and hypothesis, go through my methods, and then the results that I obtained within Brachypodium. So, copper is a very important micronutrient for all living things. Today we're focusing on plants. So as you can see, copper plays a role in a plethora of the processes within the plant. These processes include respiration, photosynthesis, pathogen response, hormonal signaling, and what I'm focusing on today is development and fertility. So most studies have been done in Arabidopsis when it comes down to copper homeostasis regulation. As we all know, Arabidopsis is a model dicot plant widely used in plant biology. We want to apply the knowledge that we have of Arabidopsis to Brachypodium, a accepted model monocot plant, so we can apply this knowledge to wheat, which is a globally important crop. So Arabidopsis is a great link with Brachypodium to the wheat model because their genome sizes are much easier to work with than wheat. So the way this regulation system works, it's all SPL7 dependent. SPL7 is a transcription factor, and it's going to work in two ways in this process. So as we can see here, SPL7 transcribes copper transport genes, and these genes transport copper to different parts of the plant, and this is when copper is available in the plant. So when copper is not available, SPL7 goes into this economy model where it activates the transcription of microRNAs that are going to degrade different proteins and enzymes that utilize copper for less important processes. So it can free that copper to be transported to more important processes such as photosynthesis. So as we can see here, our lab, the Vadamanuk lab, is trying to apply, like I said, what we know from Arabidopsis in Brachypodium. So here we have a SPL7 mutant, and we can see that SPL7 is extremely necessary for the growth and development, because without it, you see that it's heavily stunted, and fertility is also stunted as well. So the way the microRNAs work in the regulation system, they repress the genes that are using the copper for less necessary things. So the way it works is via post-transcriptional cleavage. So the microRNAs bind with components such as RISC and argonate to degrade messenger RNA before it's able to be translated into these target genes. So objectives for my study is to identify the microRNAs that are regulated in copper deficiency in an SPL7 dependent manner in Brachypodium in both roots and shoots, as well as identify microRNAs that are involved in Brachypodium fertility and responsive to copper deficiency in the same SPL7 dependent manner. And our hypothesis is that the same processes that are in Arabidopsis will be conserved in Brachypodium. So we chose two specific microRNAs, one for copper homeostasis and one for development. There are many that are involved in both processes, but we chose microRNA 172. It is responsible for floral identity and adult cell differentiation, as well as developmental timing, and we can see that it's regulated by another SPL7 family, an SPL7 family member, SPL9, as well as another developmental microRNA 156. So we also have copper microRNA 398, and 398 is known to be responsive to copper availability in Arabidopsis. We know, as studied in our lab, that it is upregulated under copper deficiency, and we also have found that it is regulated in this SPL7 manner, and that it is involved in the copper economy model that I talked about before. So the way I set up my experiment, I grew plants in control copper condition, 0.25 micromolar of copper, as well as high copper condition. We needed to grow it in this high copper condition for the SPL7 mutant to survive to the flowering stage, because it needs high levels of copper to survive. And once the flowers started budding, we transferred them to copper deficiency, so minus copper conditions, until flowering stage, where we collected the terminal spikelets for RNA extraction, which unfortunately I did not get to in the time frame of the program. So next, we quantified our results by qPCR. I did lots and lots of qPCR with these two microRNAs. So I'm going to start out by talking about microRNA 398. So microRNA 398 has 3 isoforms, A, B, and C. And we see that when comparing transcript abundance in wild type and both SPL7, that the microRNA is completely SPL7 dependent, and microRNA 398 is the least abundant in both roots and in both shoots. All right, and so now we can look at each isoform individually. You can see that I grew both control and grew wild type and SPL7 in both control and minus copper. And among all 3 of the isoforms, only isoform A is upregulated in copper deficiency in roots. And as you can see, both B and C are downregulated in copper deficiency. All right, so alternatively, in shoots, microRNA 398A and B and C are all upregulated in copper deficiency, which is important because it shows that it is involved in this copper homeostasis regulation system. Okay, now moving on to our developmental microRNA 172. So all of the 172 isoforms seem to be downregulated in the shoots. Again, we have the 3 isoforms that we are comparing. And yeah, in shoots, they're all downregulated, and they all seem to be SPL7 dependent, at least in part, while in comparison with shoots, they are not really SPL7 dependent. So when we break all of these down and look at each one individually, we see that A is the only one that's really upregulated in copper deficiency, but D is heavily downregulated in copper deficiency. And we see that it's not really that too SPL7 dependent, only isoform D shows to be partially SPL7 dependent. So, alternatively, in shoots, we see that all 3 isoforms show SPL7 dependency, which is in contrast with roots. And again, microRNA 172 is downregulated, which we saw also in roots before. B is also downregulated in copper deficiency, but isoform 172A seems not to be affected by copper availability in the environment. So, in conclusion, bring you back to my objectives, we see that for the regular copper deficiency in the SPL7 manner in Brachypodium, all 3 isoforms are upregulated in copper deficiency under SPL7, which means they all seem to be working in this copper homeostasis regulation system. And all the among all of the isoforms, MicroRNA 398A is upregulated under copper deficiency via the roots. So we see something interesting in A, but I would also like to point out that this conclusion I have here down at the bottom is consistent with what we have found in Arabidopsis. So for objective 2, the microRNAs that are also involved in fertility and both copper deficiency, of all the isoforms do gain SPL7 dependency in shoots. However, they are not dependent in roots. And we see that both isoform B and D are SPL7 regulated through copper deficiency in shoots. And isoform D is downregulated in both roots and shoots. So just to bring you back to what this looks like visually, phenotypically, we see that in minus copper, it's hard for the flower to develop. It's something morphologically wrong with the flower. So we know that microRNA 172 is involved in the pathway that's making this happen. So yeah, this is just a visual for you guys to see what's going on here. All right, so next we need to actually extract that RNA from the flowers and run qPCR on them to see the expression levels of microRNAs 398 and 172 in the flower tissues, and then go on to create CRISPR-Cas9 mutants of Brachypodium microRNA 398 and 172. And so I would like to thank my lab, the Vadamanuk lab, specifically Olena, and my mentor Miriam, and also Yulene, who I worked very closely with. I'd like to thank Cornell and BTI for having me, and also my funding sources. Okay. All right. Any questions? Yes?
How does the microRNA function to affect mRNA? Is it kind of similar to like a virus response where it double-stranded and then the plant breaks it down? Or how does that work?
How is the regulation with the microRNA work? Yeah. So the microRNA binds with components like RISC and Argonaute to break down messenger RNA. So it doesn't let that messenger RNA go on to be translated into the genes that the plant doesn't need.
Since you have been working with so many isoforms, I just wonder, for the quantification, you're looking at microRNA precursor sequence or the mature sequence?
I'm looking at the mature sequence, yes.
It seemed like one of the microRNAs was regulated by SPL7
Mm-hmm.
Mm-hmm. The other one only in the shoots, not the roots.
Yes.
So my question is, so the SPL7 must be in those tissues. If it's regulating one microRNA in the roots, how could the same gene, the other one, not be insensitive to SPL7 in the roots?
So it seems like at different stages of the plant and different parts of the plant, these microRNAs are doing different jobs and they have different targets. So they can have different targets in at different developmental stages as well as different parts of the plant. So in the flowers, it could be expressed differently because it could be doing something different or targeting a different gene. So.
Yeah, I mean, that makes sense as far as like why. I'm just wondering how they become insensitive. How does this other microRNA become insensitive to SPL7 in the roots?
Oh, I'm not sure. Yeah, I'm not sure.
Probably nobody knows.
Yeah, something to be studied.
Ask Olena.
Yes. Yeah.
Okay,
All right.
so if no further questions, Thank you again.
All right, thank you.